Bismillahirrahmanirrahim. My name is Niaz Hussain. I am from Department of Microbiology, Kohat University of Science and Technology. The topic of my presentation is cultivation of bacteria, presented to Honorable Professor Niaz Muhammad, which is Chairman of MLT. Cultivation of bacteria. What is cultivation of bacteria? A microbial culture is a method of multiplying microorganisms by letting them producing in a predominant culture media under controlled laboratory condition. What it means? It means that when we culture bacteria in a laboratory by using septic condition, microbial culture are used to determine type of organism, its abundance in the sample being tested, or both. Okay, in this slide we will discuss about purpose of culturing. That what are the purpose of culturing? Why do we culture bacteria? The first purpose of culturing is isolation of bacteria. When we want to isolate bacteria from mixed culture, so we will isolate bacteria. The second is initial step of studying and culturing bacteria is morphology and to see its identification. Another purpose of culturing is to maintenance of stock culture. The fourth purpose of culturing is to check antibiotic sensitivity. To create antigens for laboratory use, certain bacteria we have used for the use of the antigens in laboratory. We also culture bacteria for genetic study. Culturing on a solid media is another convenient way of separating bacteria in mixed culture. Okay, so how do we culture bacteria? We will culture bacteria on culture media. So what is culture media? Culture media is an artificial media that provides similar environment and nutritional condition that exist in natural habitat of bacterium. What it means? It means that culture media is provided artificially to the bacteria for its growth. Culture media mainly contain water, carbon source, energy, some trace element, growth factor, but the main thing in artificial culture media is that we have to maintain the pH accordingly to the microbe. Okay, so what are the uses of culture media? The uses of culture media is enrich the number of bacteria, select the certain bacteria that suppress other bacteria. For example, if we have a mixed culture and bacteria, so we culture bacteria and then isolate bacteria, and after that we have to suppress other bacteria and grow the desired bacteria. It also differentiate amongst the different kinds of bacteria. Okay, what is pure culture? A pure culture in a laboratory is that the bacteria are isolated or grown in pure culture in order to study function of a particular species. For example, we have a mixed culture and we isolate one bacteria, and then after that we grow that bacteria again to obtain pure culture. A pure culture is a population of cells are growing in the absence of other species or types. A pure culture may originate from single cell or single organism, in which case the cells are genetic clones of one another. Pure culture are obtained by using variety of special technique. There are certain techniques which are used to obtain pure culture. We required glassware, media, and some instrument, and these all instruments will be sterilized and used in aseptic condition to obtain pure culture. Basic requirement for obtaining a pure culture are solid media. We will obtain the pure culture on solid medium. A medium should be in aseptic condition. When we provide nutrients to single bacterium, it will multiply on that media, and it will multiply on that media and form colony. Okay, what is agar? Agar is a polysaccharide extracted from marine algae. It is used to solidify a specific nutrient solution. Unlike other gelling agent, it's not easily degraded by many bacteria. The culture media contained in a petri dish are two parts: glass or plastic covered in container. Okay, so now we will discuss about classification of culture media. On the basis of classification, the media are classified into three components. Number first, consistency, nutritional components, and functional use. Classification on a consistency. On consistency, then we again classified media into three parts. First one is liquid media, second one is solid media, and third one is semi-solid media. Cultivation of bacteria. These are the pictures of media. The first one is solid media, which contain agar. The second one is liquid media, which have no agar, and we can see here a yellow turbidity are formed. And the third one is semi-solid media. This is also some pictures. Okay, so now we will discuss about classification of culture media. On the basis of classification, the media are classified into three components. Number first, consistency, nutritional components, and functional use. Classification on a consistency. On consistency, then we again classified media into three parts. First one is liquid media, second one is solid media, and third one is semi-solid media. Okay, classification based on functional use or application. It is classified into six type of medias: enriched media, selective media, differential media, transport media, indicator media, and anaerobic media. Enriched media. What is enriched media? Addition of extra nutrient in the form of blood. In enriched media, we add some blood or serum, egg yolk, or some other components to media to make it enriched. What is the purpose of enriched media? It is used for the isolation of pathogen from mixed culture, and it also stimulate the growth of desired bacteria. When we want to grow a desired bacteria, then we grow it on enriched media. Examples of enriched media is chocolate agar, blood agar, etc. are the examples of enriched media. These are the some pictures of enriched media. Okay, number second is chocolate agar. What is chocolate agar? Agar is a non-selective, and it enriched the growth medium of fastidious bacteria. There are some fastidious bacteria which best grow at chocolate agar, such as Haemophilus influenza is an example of chocolate agar. Blood agar. What is blood agar? Blood agar contain mammalian blood. Blood agar contain mammalian blood, usually sheep, horse, buffalo, etc. And its concentration will be 5 to 10% to become enriched. Why we used blood agar? We used blood agar in order to isolate fastidious organism and to detect their hemolytic activity. Either it is alpha, beta, gamma, a different hemolytic pattern are observed on blood agar. This is the pictures of blood agar and also a chocolate agar. Uh, Number second is selective media. What is selective media? Selective media means that we add some inhibitory substances to a solid media that increase the number of colonies of desired bacteria. When we want to grow desired bacteria, we will use the selective media. Selective media and enriched media are designed to inhibit the unwanted or contamination of bacteria to help recover pathogen from mixed bacterium culture. For example, if we have a mixed culture and we want to isolate one bacteria, so what we will do, we will isolate the bacteria and streak on selective media. It will grow and form the colony. Okay, so any agar media can made be selective by addition of certain inhibitory agents that don't affect the pathogen. Classification on a consistency. On consistency, then we again classified media into three parts. First one is liquid media, second one is solid media, and third one is semi-solid media. These are the pictures of EMB and Campylobacter agars. Okay, number three, differential media. What is differential media? Differential media, certain media are designed in a such way that different between bacteria can be recognized on the basis of their colony colors. Bacteria, when we grow the bacteria on differential media, it will form the colors. On the basis of color, it will differentiate. Examples of differential media is XLD, McConkey, clad. These all are the examples of differential media. Cultivation of bacteria. These are the pictures of McConkey and clad agar. We can see in this slide the growth of pathogenic E. coli on clad agar, which is in bluish color. Transport media. What is transport media? Transport media is used for the transportation. When we isolate and we take sample from a human being, from any persons, so then we have to transport it to laboratory, carry to laboratory. So for that we used transport media. Transport media should be fulfill the following criteria: temporary storage of specimens being transported to laboratory for cultivation. It maintains the availability of all organisms in the specimen without altering their concentration. Transport media contain buffer and salts. It lack the carbon, nitrogen and other growth factor. Why? Because to prevent the microbial multiplication. If transport media have carbon, nitrogen and other growth factor, then what will be happen? Then the microbe will be multiply and the desired product will be not obtained. Examples of transport media is there are different transport media, but main is carry baler media for Campylobacter species. And the another example of transport media is Sturz media. This is the pictures of blood agar and also a chocolate agar. Indicator media. What is indicator media? Indicator media means when it change its color when bacteria grow in it. For example, if we grow bacteria in a media and it change its color, so by changing its color we can identicate the bacteria, that either this is gram positive, gram negative, fastidious, non-fastidious. Example, the example of indicator media is urease media. These are the examples of transport media, and the another example is of indicator media. These are the pictures of EMB and Campylobacter agars. Anaerobic media. What is anaerobic media? Anaerobic media is used for the growth of anaerobic microorganism. Anaerobic media, anaerobic bacteria need special media for growth because they need low oxygen content. So for that the anaerobic media is designed. The example of anaerobic media is thioglycolate medium. These are the pictures of anaerobic media, mainly thioglycolate media. These are the pictures of McConkey and clad agar. These are the examples of transport media, and the another example is of indicator media. Streak culture. It is used for isolation of bacteria in a pure culture from a clinical specimens. A platinum wire is used, and one loop full of the specimen is transferred on the surface of dried plate and then spread all over the plate. The inoculum is then distributed thinly over the plate by streaking it with loop in a series of parallel lines in different segments on the plate. This is the example of streak culture. In picture we can see one is from left to right, and then it is primary, secondary and then tertiary. And the another we can say technique in streak culture is that we have to do from head to tail. This is the clear picture of streak culture, and we can say the primary culturing, secondary and tertiary culturing. Lawn culture. It provides the uniform surface growth to the bacteria. Lawn cultures are prepared by folding the surface of the plate with a liquid suspension of bacterium. Why we use lawn culture? For bacteriophage typing, for antibiotic sensitivity testing, and different preparation of antigens and vaccine, etc. We used lawn culture. These are the examples of lawn culture. In a plate, we can see a white antibiotic discs are present, and the bacteria is inhibited. Their growth are inhibited by antibiotics. Straw culture. Straw culture are made in tubes, either in slant forms, either in from top to bottom. What are the uses of straw culture? It provides the pure growth of bacteria from slide encapsulation and other diagnostic tests. For diagnostic test, we used straw cultures. Transport media. What is transport media? Transport media is used for the transportation. When we isolate and we take sample from a human being, from any persons, so then we have to transport it to laboratory, carry to laboratory. So for that we used transport media. Transport media should be fulfill the following criteria. Okay, in this slide we will discuss about purpose of culturing. That what are the purpose of culturing? Why do we culture bacteria? The first purpose of culturing is isolation of bacteria. When we want to isolate bacteria from mixed culture, so we will isolate bacteria. The second is initial step of studying and culturing bacteria is morphology. Pore plate culture. What is pore plate culture? Inoculum is added in Malaton agar mixed with and pure sterile petri dish. Allow it to set deep of the medium. What are the function of the pore plate culture? Give an estimate of viable bacteria count in a suspension. It is used for quantitative urine cultures. This is the picture of pore plate method and how we can perform this. This is an overview of a pore plate method. Spread plate method. In this technique, we first put the sample on plate, then it is spread by the help of glass spreader. Then incubation is occur. After some time, surface colonies are formed, and then pore plate method is performed, and then what sterile media is added and mixed well with the inoculum, and after some time, and after 24 hours, the surface colonies are formed. Thank you so much for your precious time. If there is any question regarding this topic, so you can ask in comment section and I will be available to answer. And once again, thank you so much for your time.